Dear viewers, welcome to my video lecture. Topic dark reaction. Now, let's see about the definition of dark reaction. Dark reaction is also called as a Calvin cycle, C3 cycle. C3 cycle is defined as a set of chemical reaction performed by the plant to reduce carbon dioxide and other compound into glucose. Calvin cycle is also known as a C3 cycle or light independent or dark reaction of photosynthesis. This Calvin cycle takes place mainly in leaf of the green plant, more specifically in the stroma of the chloroplast. This plant that produce energy from Calvin cycle in the presence of light are known as a photoautotrophs. Why we called dark reaction as a C3 cycle means because it contain first stable compound. It consists of phosphoglyceric 3 carbon compound. So it's called as a C3 cycle. And it is also called as a carbon fixation. It occurred in this cycle, so it's called as a carbon fixation cycle. And this dark reaction is also called as a Calvin cycle. It was discovered by the scientist Melvin Kelvin. It's named after the scientist. This Kelvin cycle is divided into three stages. The first stage is carbon fixation, second stage is reduction stage, and the third and final stage is regeneration stage. First, let's see shortly about this three classification. The first one is carbon fixation. It is a process wherein photosynthetic organisms turn inorganic carbon into organic compound. Carbon dioxide fixation is a type of carbon fixation where the carbon dioxide from the atmosphere is converted into carbohydrate. It is the first key steps in Kelvin cycle. This carbon fixation process occurred during the light reaction phase of the Kelvin cycle because it requires light to complete this segment of the cycle. In this stage, 5 carbon compound ribulose bisphosphate is joined with the carbon dioxide and produce 2 molecules of 3 carbon compounds phosphoglyceric acid. The second stage of Calvin cycle is a reduction stage. The 3 phosphoglyceric acid molecule created to the carbon fixation are converted into molecule of simple sugar, glucose. This stage obtains energy from ATP and NADPH formed during the light dependent reaction of photosynthesis. Calvin cycle become a pathway in which plant convert sunlight energy into long-term storage molecule such as sugars. The energy from the ATP and NADPH is transferred to the sugars. This step is known as a reduction since electrons are transferred to 3 phosphoglyceric acid molecule to form glyceraldehyde 3 phosphate. In this stage, 2 molecule of phosphoglyceric acid is reduced as glyceraldehyde 3 phosphate. Glyceraldehyde 3 phosphate contain 2 molecules. The third stage is a regeneration stage. Regeneration is the third stage of Calvin cycle and it is a complex process that requires ATP. In this stage, glyceraldehyde 3 phosphate molecule are used to produce glucose. This is known as a regeneration because ribulose bisphosphate that begins the cycle is regenerated from glucose 3 phosphate. Simply, this is the process of regeneration of ribulose bisphosphate. Now let's see this Calvin cycle briefly. At the first stage, carbon dioxide acceptor is ribulose bisphosphate. For our convenience, we take 3 molecule of RUBP at first step. It should be considered as 3 Calvin cycle. 3 molecule of ribulose bisphosphate is fixed with 3 molecule of carbon dioxide. When this 3 ribulose bisphosphate and 3 molecule of carbon dioxide are joined together, we have to get 3 carbon compound. but this is highly unstable. So it breaks down and produce 2 molecule of phosphoglyceric acid in this reaction. The conversion of ribulose bisphosphate into 3-phosphoglyceric acid is a carbon fixation stage. At the second stage, 6 molecule of phosphoglyceric acid is phosphorylated for this reaction. It needs 6 ATP and ADP, and it gets 6 molecule of 1,3 bis-phosphoglyceric acid. This number represents the position of phosphate group. At the next step, this 1,3 bis-phosphoglyceric acid is reduced into 6 molecule of glyceraldehyde 3-phosphate. This reducing agent are 6 molecule of NADPH2 into 6 NADP or oxidized. This 6 molecule of glyceraldehyde 3-phosphate is gained as 1 molecule of glyceraldehyde 3-phosphate. The reduction of phosphoglyceric acid into glyceraldehyde 3-phosphate is a reduction stage. Now let's see about regeneration of RUBP. After that, we have other 5 molecule of glyceraldehyde 3-phosphate is continued in this cycle. 5 molecule of glyceraldehyde is converted into 3 molecule of ribulose 5-phosphate. In this step, 2 inorganic phosphate are released. This 3 molecule of ribulose 5-phosphate is converted into 3 molecule of ribulose bisphosphate by the addition of 3 ATP. The conversion of glyceraldehyde 3-phosphate into ribulose bisphosphate is the regeneration of RUBP. For 1 Calvin cycle, we utilize 3 ATP and 2 NADPH2. Now let's see about product of Calvin cycle. 1 molecule of carbon is fixed at each turn of Calvin cycle. 1 molecule of glyceraldehyde 3-phosphate is created in 3 turns of Calvin cycle. 2 molecule of glyceraldehyde 3-phosphate combine together to form 1 glucose molecule. 3 ATP and 2 NADPH molecule are used during this reduction of 3-phosphoglyceric acid to glyceraldehyde 3-phosphate in the regeneration of RUBP. 18 ATP and 12 NADPH are considered in the production of 1 glucose molecule. This is a reference slide, and thank you so much for your listening.